thanks for coming. If anyone's not on the Peaty Pals mailing list and would like to, you can just thing here, just stick your email address down there. And if anyone knows anyone that can't actually get to Peaty Pals but would like to come, that's maybe stuck in their house or is out in camp or something, let me know, because I can always go around and help them with teams or something like that. So yeah, this week we're really lucky to have Lydia. She's about to leave, so lucky us for getting her just in time. Lydia's going to tell us all about kelp, and she's been working, well, you were studying with the University of Hull and also with SAERI and BATS whilst you're here, weren't you? So yeah, and we've done work from what I can gather all around the world, and looked into what kind of things in the sea like to eat kelp when it's degrading, and about how kelp sequesters carbon, and worked in Namibia and Madagascar, so quite a CV. And I'm really excited, like when you see the kelp on our beaches and you see that kind of those huge big bits of kelp and tiny little holdfasts and how they can hold on in all our storms. I think it's a fascinating seaweed to study, and yeah, it'd be great to hear more from Lydia. So thanks very much for Lydia. We'll probably, if you have a burning question, go for it in the meeting, but otherwise we'll do questions at the end. So there's a couple of people online that might want to have questions. So, yeah, if you need, there's a seat here or a couple more at the back if you want to grab a seat, and one here. I don't mind either.
So, as Bryn touched upon, I'm working on kelp in the Falkland Islands. I just wanted to say a massive thank you for inviting me to talk. I'm now in my final year of my PhD, which has absolutely flown by. It's pretty terrifying, to be honest with you. But I'm just going to talk through the basis of my project and where I'm at now. I hope you enjoy it. So, first of all, I just want to say a massive thank you to a few people. So my supervisory team, who have helped on the way, SAERI for hosting me and organising all of the logistics, and then most importantly, I'd like to thank the Falkland Islands community. I've had a lot of wacky requests and for people's space and their time, and people have constantly gone out of their way to accommodate me and support me with my project. So I really appreciate that, and I will genuinely, seriously miss the community here in the Falklands. So thank you. So just to start with, I'd like to discuss what kelp actually is. So kelp is a macroalgae. So it's photosynthetic. It is not a plant. It's actually very distant evolutionary-wise to green plants. It's also very distant genetically to other macroalgae like red algae and green algae. It is very, very diverse in its genetics and its morphology. Even here in the Falklands, with a single population, there's high levels of genetic diversity. On this image here, I couldn't find one for the Falklands or the South Antarctic, sadly, but just shows you the level of diversity we see in kelp around the world. This is Macrocystis, one of the species you guys will see a lot of in the harbour and everywhere else around the Falklands, to be fair. So yeah, bit of a background on kelp. It covers 25% of coastlines globally in temperate to subantarctic regions. The reason kelp is important, which is the reason that I absolutely love it, is it's an ecosystem engineer, and essentially what that means is it provides a habitat for other organisms, so it forms the base of the trophic food web. It also provides food, and this structure of kelp is extremely heterogeneous. This is a diagram of a kelp from the UK, and it shows the distribution on an individual kelp across an individual. So here we've got the fronds, where you've got about 16 species. Then you go down to the stipe, and you've got quite a lot of epifaunal, so it means that they grow on the kelp. They're often sessile. So you have about 122 species within this area of the kelp itself. And then you move down to the holdfast, which supports 237 in this study. And that was kind of what I wanted to look at in the Falklands, is the holdfast. Because when we look at kelp forests in general, we're looking at the whole ecosystem, and we never really go into an individual kelp and the provisions it provides itself. So that was the knowledge gap that I was most intrigued with. I have to get back. There might be a bit of that. So what does kelp do? Obviously, it is really important for biodiversity, which is inherently good because it supports a resilient ecosystem, especially in a changing climate. But it's also very beneficial for humans. So when we're looking at ecosystems, we look at goods and services and how we can quantify the importance of an ecosystem to us as humans. So kelp forests are valued at $500 billion per year, and they think that is a slight underestimate as well. The main ways in which it provides ecosystem services is through coastal protection. So kelp provides a buffer for wave action. We're seeing an increase in storm incidence and intensity, so having this buffer to protect against coastal erosion is really, really vital at the moment. It's also potentially really useful in terms of the carbon cycle and carbon storage. Kelp itself does not have a root system or directly sequester carbon. However, there are pathways for kelp to potentially store carbon long term. However, there is research showing that negative feedback loops in the changing climate with warming oceans and ocean acidification may alter this, so the benefits of kelp as a long-term carbon storage resource are reducing, which is sad. Then agriculture, in a lot of communities globally, they will harvest kelp, so obviously you can get the Davilia in Chile, they harvest that and eat the stipe of it. It's often done in a way that removes the whole kelp, but they're looking at ways to keep some of the kelp now so that we can maintain that biodiversity. But it's a great food source, and they're also looking at it for using it to make alternatives to plastic with the alginate, which is quite exciting. And then fisheries, one of the most important ones is kelp support biodiversity. They also act as a nursery. They've got really nice, safe, sheltered environment. They can hide from predators. So we have, often they're called like a nursery. So you'll have smaller or larval fish or commercially important fish or prey for commercially important fish will be in the kelp forest, but because it's a very connected environment, there can be like an exchange between the pelagic and the coastal zone, supporting sustainable fisheries and fish stocks. And then finally, tourism. So I think diving is obviously increasing. We're looking at more tourism to Antarctica. Kelp forests at the base of the food web support everything above it and the megafauna that people come to see. So when we're honing in on the Falklands, so fisheries represent 58%, 53% of the Falklands GDP, and kelp forests are vital in sustaining the commercial fisheries here in the Falklands, and tourism, as I just mentioned, is increasing. So having healthy, pretty kelp forests with nice clean water and lots of megafauna is really important in the Falklands in particular. There was a study by Bailey that found that kelp forests here in the Falklands are valued at 3.24 million per kilometre squared per year. So that's quite insane when you think about it. So what's happening to kelp? It's a lot less talked about than coral bleaching or the loss of seagrass meadows. It's not as charismatic, but I think that is changing. But as you can see, the picture looks quite bleak. We're seeing global decline in kelp, having compound effects of various different factors that I'll get into later. But the overall story is we're losing the kelp, and that is causing a loss of resilience in habitats globally. And as you saw previously, kelp covers a lot of areas and supports a mass of biodiversity. So this is really important. So what's driving this? Oh, hang on. It's a fun graphic I made. So we've got our nice little biodiverse kelp forest. Then if we lose them, just to give you a little bit of a picture, you lose the biodiversity, there is less wave attenuation, you've got a much flatter, homogeneous environment. So you'll see, we call it like a regime shift, which ultimately affects ecosystem resilience, loss of goods and services, and just loss of biodiversity, which we don't like. So what are the threats to kelp forests? Most importantly, I think, is climate change. Kelp are found to struggle with warming oceans and ocean acidification. Their respiration rates go up and they are less resilient to other stresses such as storms. Overfishing, when you remove top predators, what other individuals or macroinvertebrates can become more abundant and compete with the kelp for space, which then there's a reduction in abundance. Invasive species, along a similar line, you're altering the ecosystem and its equilibrium and changing the competition. Pollution, turbid waters, dirty waters, all alter the metabolic processes involved in kelp functioning. And here in the Falklands, you've got beautiful, pristine, clear water outside of the harbour. So kelp thrive in that. Increased marine activity and human activity within the water stirs up the sediment, reduces the capacity for kelp to photosynthesize. Then storms and disturbance events such as marine heat waves also add another level of stress. But I think the overarching story is that it's not one thing causing decline, it is the compound effect of a mass of different stresses which alone can be tackled, but we can't start tackling them alone anymore. But what about the Falklands? So this graphic shows whether there's been a decline or an improvement in kelp forest health globally. So blue shows the improvement, the size of the bubble shows the percentage of the improvement. Here in Patagonia, you can see that actually the kelp forests are thriving. They are some of the best in the world. But why is that? So the Falkland Islands has a offstream of the Antarctic circumpolar current. This is a cold, nutrient-rich upwelling system. So it's keeping the water cool, it's keeping the nutrients flowing, and it's keeping the kelp pretty happy. It's also, the kelp here is sat at what we call the leading edge of its population. So it's on the poleward edge of its distribution. That means that its thermal tolerance, if you're looking at it latitudinally, is able to go a lot higher. So a warming climate isn't a massive pressure right now for kelp, and also the organisms associated with it. Then I think one of the main key points is it's quite far away from people. So we've got limited pollution, very clear water, nice nutrient-rich water, and fisheries that are pretty well managed, so we can keep that ecosystem healthy with lots of nice top predators, which, if you're looking at other areas where we've seen phase shifts, such as California, where they lost 90% of their kelp, it was due to a loss of apex predators, mainly due to overgrazing by urchins. But how stable is the situation? I think Paul can probably agree it's relatively stable for now. However, the climate's changing pretty rapidly. So what we want to understand is whereabouts in in the ecosystem health is it. So this is a stable and resilient ecosystem. Here is where we start to see a loss of resilience, and then once you go past that, you can see a phase shift. So go to an urchin barren or a turf reef. So what I would like to know is whereabouts within this are we, and why are we in it where we are, to understand and predict future changes to the climate. Back over here. So that's where my research fits in. I'm also talking very quickly. There's three core aims of my project. First one was a comprehensive assessment of kelp holdfast micro-ecosystems in the East Falkland Islands, honing in on two species of kelp that are buoyant. So the Vilia antarctica, which is the southern bull kelp, common name, and Macrocystis peripheral, which is the giant kelp. So mainly looking at this holdfast structure that provides the complex habitat. Then I wanted to look at the risk of disturbance events on the ecosystem and its resilience, and looking at how likely it is that a disturbance event, an intense disturbance event, would alter the ecosystem here in the Falklands. And then finally, I wanted to look at the Falkland Islands kelp and its role within the Southern Ocean as a whole, in terms of connectivity, but also potentially the transport of non-native species with changing oceanographic features. So this is over here. This is what I've spent the last 3 years looking at. So this is the holdfast structure of Vilia. It's a discoid morphology, and I think when you're looking at it from above, it looks pretty homogeneous, not like it's going to support much, and it should be solid. However, you get boring animals such as amphipods and isopods that start to burrow their way in and create holes. Once those holes are created, you have a succession and a full community built. So this is a CT scan, thank you to the hospital, of a holdfast structure. As you can see, you've got these holes and then there's burrows, and it's actually really complex under there. We've even got CT scans that show inside, without any way of escape, limpets that appear to have just got in there when they were small and then have been able to sustain themselves just on what's in there alone, which is pretty exciting. And then when you compare this to the other kelp holdfast structure I'm looking at, which is the Macrocystis, it's much more root-like, it's very tangled, very messy, and very slimy. It traps sediment, but it looks a lot more complex. But interestingly, I found higher levels of abundance in the Vilia holdfast than any of the Macrocystis. So in one holdfast about this size, I found 800 and something animals, individuals, which is pretty shocking when you're looking at it. It just looks so innocuous as a part of the kelp forest. So I think that's incredible, just how small it can host so much. So how I went about assessing the biodiversity, I found some sample sites for the Vilia that were accessible. So Vilia is situated in the intertidal on the rocky reefs, and then I take a hammer and a chisel and hit away at them and then put them directly into a mesh bag. They are glued on there, so it's a pretty intensive process. And then SMSG very kindly donated their time and their divers to collect Macrocystis holdfast for me, as they are subtidal. After that, I go through and I pull off, this is a cross section of the Vilia, pull off all of the animals. So if you look at this, I don't know if you can see, that's sediment, but when you start going through it with your tweezers, you'll probably find about 20 different amphipods in there at least, in that little section. So it's quite time consuming, and I started seeing amphipods everywhere. It's quite hard when you're trying to eat. And then pull it all off, pop it all into petri dishes. I try and split by morphospecies, so that's what that's doing. And then I send them all to specialists because I'm not quite confident on knowing the species for however many thousands of animals I have. So I'm working with collaborators globally who have received samples from the Falklands and are looking at them as we speak. So hopefully we'll get a much clearer idea and much more precise idea of the biodiversity here in the Falklands. So that's that bit. So some preliminary results. This is from my first field season, and I want to stress this has not been based on stats. This is just eyeballing what I've seen with my own 2 eyes. So the macrocystis that I collected intertidally was from Stanley Harbour. I made that mistake once, and I'm not doing that for further replicates. But in there, I found lots and lots of snails. A lot of this crab here called Acanthocyphus albatrosis, or the fighting crab. Then some fish and a lot of the Asterina fibras, which is the bat star. Then when you move to the cleaner, deeper, more exposed waters, you see the community kind of shift. You get a lot more of the rough brittle star, the pink sea urchin, limpets, and then a lot of predatory worms. So there is quite a stark difference just by looking at the ecosystems. And then similarly with the Davilia, so I looked at 2 sites, Surf Bay and Hooker's Point. So Hooker's Point is a lot more exposed, rocky surface. The Dolphin Beach is sandy in most places with a few rocky outcrops. And again, I found very different communities within such small spatial scales. In the low exposure site, I found much more like spread with lower abundances of all organisms, but more limpets and crabs and larger organisms. Whereas when I moved to the high exposure, you see thousands of small, boring amphipods and isopods. This one here is called Parawaldechia kiddery, and I've been probably counted over 10,000 of those alone so far. So there's a very big difference in the community structure. Hopefully we find significant results, or maybe not, but yeah, that's the preliminary results for that. So I think that tells us a bit about the biodiversity we are hosting here in the Falklands, and how even on an individual kelp plant scale, there is mass of biodiversity, which is incredible. It's really exciting to see. It gives us a baseline for us to monitor going forward to quickly spot changes in the environment. One thing that we have found is this anemone here, which is invasive in other areas of the world. It's called Metrodinium sina. It outcompetes with mussels and barnacles for space on the shore, which has had quite a knock-on effect on commercial fisheries in other areas globally. So I think that just highlights that whilst the ecosystem here is thriving, it's rich, it's abundant, there are still threats, and it's important not to ignore them because we don't want to end up with a homogeneous ecosystem. So moving on to the second part of my project, we understand the biodiversity, which hopefully in a little bit of time I can look at the ecosystem functioning and assess that. So then I want to look at how a disturbance event would impact this potentially. So a disturbance event is a discrete event that is acute in nature. So we're talking like a storm, or in my case, a marine heat wave, whereas when we're talking about stress, it's chronic, so water pollution or warming seas. So I was looking at marine heat waves. This is what a marine heat wave looks like. It's categorized as an event of prolonged abnormally high ocean temperatures, which is pretty vague, but we're going with it. So when we're looking at it, it's got this slow incline, then it rockets before dropping back down, which is important for my experimental design. Now, whilst the Falklands doesn't currently experience lots of marine heat waves, recent research has predicted that they're going to increase in both intensity and periodicity over the coming years. So there was a paper recently out that suggested that we could see 3 to 4 instances of marine heat waves per year, reaching a period of a maximum of 10 days at this peak. and the peak being 10 degrees warmer than the current sea temperature. So, as I mentioned earlier, we're at the leading edge of the population for most of the species here, which means that they are comfortable in the cold. But populations within a species can display different thermal tolerances. So whilst they're in the same species, their thermal tolerance range may be different. So whilst they may be able to tolerate warmer water because we're at the leading edge, it also may be plasticity within the populations. We don't know. So this is what I looked at. I looked at the crab, and I wanted to look at where its range was for the Falkland Island population. So here we have a nice little graph of thermal tolerance. So you have the generalist here, which shows it's comfortable living in a broad range of environments. Then you have cold tolerant, warm tolerant, and specialist. So my predictions are that the crab would sit on the cold tolerant end of the spectrum, which means it could be potentially more susceptible to thermal stress. So this is the crab that I studied. Why did I pick this crab? So it's an intertidal predator, and as I mentioned earlier, apex predators and predators in general are really important for maintaining a healthy and resilient kelp forest because they eat things that compete with the kelp and control populations. So this crab, so you can see it's got one really massive pincer. It's called the fighting crab. Its diet is largely mussels, so it will go and crush mussels. This creates space. The space could be taken up by kelp, supporting, again, a healthy ecosystem. So how did I test the thermal tolerance of a crab? I was looking at its fitness rather than physiological parameters. And when we're looking at fitness in ecology, we're looking at how much energy an individual has to expend. So what's their energy budget? Because every individual has a set amount. And when an animal is under stress, it will direct more of its energy into survival over growth and reproduction. And there's various ways you can measure fitness. I mean, one example would be the beat test in humans, and I essentially conducted that on crabs. So I used dorsal ventral rising reflex to assess their fitness. It sounds fancy. I flip them over, I video them flipping over, I time how long they take, and I repeat it 5 times. I repeat it 5 times to look at recovery rate as well. So when the crabs flip, time it, give them a little bit of a break, and then I keep on doing it. In terms of experimental design, probably should have gone for that first. This is what it looked like. A lot of polystyrene boxes and a lot of plastic, but it was about 12 months of planning, so be gentle. So this is the overview of my cabin up a hillside. So I had 5 tanks of treatment, 5 tanks of control. Each tank had 6 crabs in it, and then I had all of the animal husbandry stuff, homemade aircon unit to keep the temperature as low as possible. And then how I designed it was to try and mimic what we see here. So at the control, I kept the temperature as stable as possible in a non-temperature controlled environment. And then for the treatment crabs, I kept them at the same temperature for 7 days, getting a baseline of what their fitness was previously. Then I increased the temperature by 2 degrees every single day for 5 days to mimic that peak. Then I kept them at plus 10 degrees, so about 21 degrees for 5 days, and then dropped them back down for 5 days, and then took a experimental reading as well to look at whether, if there was a change in performance, was that recovery quick, was it slow? So for each crab, I gave them a 3-minute period to perform their flip. If they weren't able to perform within 3 minutes any of the flips, it was recorded as a fail. So what I saw, again, not statistically significant, visually, smaller crabs seemed a lot more resilient to thermal pressure, with the slower crabs showing an obvious decline in flip time over the course of the increase and the peak. I also fed them all 3 grams of squid each, which should have been absolutely plenty. In the first feeding, they all ate around 0.3 grams. In the feeding, during the peak, the crabs under treatment ate all of the squid. So hypothetically, that could indicate increased metabolic requirement, because they're trying to maintain a more stable environment in themselves, like homeostasis. So that's all observational. What I did also see was, once I dropped them back down to baseline, it appeared as though all the crabs would return to the same performance capacity, with variation between gender and size, but in trend with their original baseline. So whilst it looks like there was a decrease in fitness during the marine heat wave, it's potential that there was recovery as well. But this is important because whilst flipping doesn't seem interesting, if a crab's unable to flip itself back over, it's unable to avoid predators as much, it's unable to feed because it's probably got lower strength, and feeding takes a lot of energy. So whilst the flipping is non-lethal, it suggests that potentially their fitness and their survivability during disturbance events is decreased. But again, all hypothetical. So we've looked at the Falklands as an ecosystem and the biodiversity and the resilience and the function of it. Now I want to talk about how the Falklands fits into the global, more specifically the Southern Ocean picture. So the term the Kelp conveyor belt is discussing kelp rafts. So the 2 species I chose to look at are buoyant. Macrocystis has pneumatocysts on its blades, so gas bladders, and Davilia has in its blades, it has a honeycomb structure, so it looks really thick and really leathery, but it's actually quite spongy. This means that they're both buoyant, and they're both really thick, really slow growing, and quite tough. So you have a storm event, for example, comes, wipes off some of the standing cover of the kelp, it then rafts. Where it rafts, who knows? Some people might know, but the level of rafting is debated and it's still trying to be consolidated. In 2002, someone estimated there are 70 million kelp rafts in the Southern Ocean. That was based off 2 transects in one area. So is the number that high? I don't know. However, kelp rafting is a significant factor, as is shown by this study here, which looks like a lot of circles, but it's actually interesting. So this is showing in this one the genetic diversity of the isopod, the Norius stevensi. It's a long little yellow one that I found a lot of in the Davilia. This shows really low levels of genetic diversity across tens of thousands of kilometres. So it suggests that despite these massive spatial barriers, there is something maintaining population connectivity. So there are reproductively viable organisms choosing or having no choice to remain associated with the kelp raft. Similarly, for the parallel echia, which I mentioned earlier, again, really, really low genetic diversity. Does that mean that rafting is a choice by these organisms, or is it just that they happen to stay on the raft and it's happier staying there than dropping off once it's rafting? To be decided. Whilst connectivity is really important for maintaining genetic diversity, which is great for preventing disease and keeping the ecosystem healthy, in the changing climate, the trajectory of the kelp rafts is also predicted to change. So we're seeing an increase in storm-driven eddies, so swells in the ocean during the Antarctic circumpolar current. Previously, the Antarctic circumpolar current acted like a cricket bat, batting everything away and keeping Antarctica as its own ecosystem that was very endemic. However, we're seeing these storm-driven eddies increase in intensity and reduce that physical barrier to transportation and connectivity between the subantarctic and the Antarctic. We also have research that has found that kelp rafts have been washing up on Antarctic islands, which suggests that this is probably pretty accurate. When you couple that with the depletion and retreat of ice sheets in the Western Antarctic Peninsula, making more coastal space hospitable, there is a threat that non-native species could be transported, well, native to us, could be transported to Antarctica and be introduced and successfully established, with Antarctica warming rapidly, the reduction in physical barriers and the alleviation of the environmental barriers to successful establishment. Here is a particle tracking model, I should have cropped it, of South America and where the kelp was predicted to go. So this shows that the particles are expected, so the kelp rafts, if they are lost here, expected to go round and round before they reach West Antarctic Peninsula. That is about 18 months of travelling in the ocean. So you would expect that most things would jump off or die, and that would be that, so reducing the risk. However, my research was looking at exactly that. This is what I hypothesised, that mobile species would immediately abandon the ship. Then, as it rafted over time, other things would drop off, potentially because they were dead or because the kelp had deteriorated. I tested this with Davidia at Fipas, floating laundry bags full of kelp, the kelp holdfast, that I changed periodically, and then I, at the end, collate all of the biodiversity information to get my total community structure. Then over time, as things drop off, I remove those from the community composition. And the stats, these aren't accurate, but this is what it should look like once it's done, is I'm going to look at 0 to, this one was 20 days, how did the community change? Are there trends in the things that are jumping off, and why are there trends? Do they share different ecological traits? Are they more sensitive to changes in the environment? However, very, very preliminary results suggest that actually, whilst there is an immediate exodus, the exodus does not trend with one species showing that behaviour for Davidia. But I also conducted a long-term rafting experiment out at Urchin Rock using macrocystis. That was rafted or simulated rafting for 16 months, so almost the 18-month benchmark of reaching the West Antarctic Peninsula. And here on the right is everything I pulled off a holdfast after 16 months. Largely worms, but you can also, you can't see, you can also potentially see that there are crabs and other mobile species. Some of the crabs were young and small, suggesting that this ecosystem is still actually operating as an ecosystem post-attachment, because you've got to remember that there isn't actually any requirement for it to be on the rock, but the environment, increased wave action, increase in UV irradiance, all may affect the community. But aside from that, the structure is still the same, and it is providing food for them as well. So that's really exciting. Still got a lot to go through. That whole part of that one came off. Took me about 16 hours in total to remove all of the animals. So it's a lot of work, but hopefully that's going to start providing an indication of what we can expect to happen in a changing climate, and whether these organisms that here doing some MaxEnt modelling, so looking at whether they would be capable of surviving in the West Antarctic Peninsula or South Georgia, are they potential invaders? by using like environmental variables and then known thermal tolerances, you can just get a nice little graph with colours. Red means no, green means yes, it could potentially survive that if it was introduced. So I think that'd be a really interesting way to take the project. So what's next? I don't know how long I've been. I'm hoping that this research provides a comprehensive description of kelp holdfast micro ecosystems that would hopefully provide an early detection system for the Falklands and monitoring the ecosystem here and keeping it as pristine as possible, because there are very few places left in the world that are these days, so I think we should protect it. Hopefully it will inform sustainable management of fisheries and marine spatial planning, and then just add to our understanding of Southern Ocean biogeography, and help us more accurately predict what's going to happen in a changing climate. Thank you.
What a lot of work. We'll just see if you've got any questions online, and then if not, hi, Heather, if you can hear us, have you got any questions for Lydia? If not, we'll go out to the room. Hi, Heather.
She's gone to sleep.
We'll carry on. Heather, if you've got any questions, just shout out. Thank you. Anyone else got a question?
Have any of those rafts that the predicted path of around the Antarctic, have those been tracked with any kind of devices so that?
So that was done using computer-based modelling, but I think it's something that people are looking into, and I did actually discuss potentially doing that, releasing some rafts with GPS trackers here in the Falklands, because I anticipate that there is a high level of retention, because when you walk on the beaches, you see a lot of very fresh kelp with a very similar ecosystem to what's on it when it's in situ. So the level of retention, I think, is something that hasn't been assessed properly, so it challenges that, yeah.
So one of the things that I was thinking is if you were able to track them, then it would be more, instead of having your experimental area where you then check the biodiversity after what period of time, but it's in a set place, like you did there.
Yeah.
If you were able to track them and check them, then that would be more.
Yeah, I absolutely agree. I think I did consider, if I had time this season, doing an immediate assessment without using the bags. However, because most of the holdfasts have very different structures and size affects it, being able to backtrack and see what was on it originally would be quite difficult. But I think it'd be interesting. One of the things they're doing in New Zealand is using genetics to look at where the rafters come from. So that's another way they're trying to explore, where things are arriving from. And I think there was some from non-New Zealand locations, which they used the amphipods actually to look at where it arrived from, which then informs the computer models.
Yeah, I think Kath and Hugh put in a grant to look at exactly that, putting satellite trackers on. I don't think it was successful, but they're still trying. It's a good idea.
Just find the money.
Yeah, that's right.
Hi Lydia, thanks very much for the super interesting talk.
Yeah.
Two questions related to each other, but you talk about the leading edge of favourable sort of habitat or favourable conditions for the kelp. Do you have a feeling for what speed that's moving? And then secondly, you suggested there might be some useful applications in the management, in local management, things like marine spatial planning. Do you have any sort of thoughts on what that could be?
So with the leading edge question, it's less at how much it's moving. You do see a contraction with populations as they are on their trailing edge, as it gets too warm for them. You see this squeeze towards the poles. I mean, the Falklands is pretty sheltered from rising sea surface temperatures because of the cold current that comes through. So I think it would be hard to say, but I was expecting the crabs to show like a population specific trait of not being tolerant towards warm water, but it didn't seem to be the case. And then with management, it's a tough one. I haven't gotten that far into it yet, but I think with a lot of kelp forest stuff, there's only 3%, I think, of kelp forests globally that are protected in any kind of format. 3 to 60, 2 different sources said 2 different things. But I think it would be maybe looking at, you know, minimizing traffic around kelp forests that are away from things, because in the Falklands, like if you go around the islands, I'm pretty sure you mentioned this, there's really high genetic diversity of the kelp. So just protecting a proportion of all of it would be nice. But I don't know enough about that, sorry.
Okay, thanks.
Any more questions? I've got one. What's your favourite little creature of all the things that were living on the planet? Do you have a favourite?
Well, I used to absolutely hate the worms because they you pull them out and they flick in your faces and they're like disgusting. But I had the opportunity to go meet Teresa Derbyshire, who wrote her PhD on worms of the polychaetes of the Falkland is tidal, and sitting there with her for 2 days and going through them, I actually found them quite pretty. And it's like appreciation for something I really didn't like before.
These are the worms then. Brilliant.